Everyone, thanks for sticking around to the end of the day for this very special jointly hosted seminar between the Department of Wildlife and Fisheries Sciences and the EEB programme. Today we have the pleasure of listening to Dr. Carol Baldwin, who is the Curator of Fishes and Department Chair in the Department of Vertebrate Zoology at the National Museum of Natural History, Smithsonian Institution, Washington, D.C. Dr. Baldwin asked me to keep this brief, and I usually try and make a silly joke or something like that when I introduce people, but I'm not going to do that today. All I'm going to do is tell you that Dr. Baldwin told me personally that she gets many requests every year to come and give seminars, and she only gets one a year. So we're very lucky to have her with us here today to listen to her research about deep reef fishes and other animals in the Caribbean.
Thanks, Kevin. Well, this is my first trip to Texas A&M, any of the campuses, so it's an honor to be here, and I just want to thank the graduate students for inviting me. I've really had a fun time today talking to everybody and learning about what you're doing and seeing so many young faces in here. We don't get that so much at the Smithsonian, and a few postdocs, fellows here and there, so this is really fun for me. So I want to tell you today about some research I've been doing over the past 8 years using a submersible to explore deep reefs. I don't know, do we want this light off, so would that be better? Prior to this, I spent most of a decade basically doing this, getting an airplane ticket to someplace in the Caribbean or Western Atlantic and putting on scuba gear and collecting what we call cryptobenthic reef fishes in the Caribbean. These are small, bottom-associated fishes. And the reason we were doing this is that in the 2000s, in addition to traditional anatomical data, we were able to integrate that with new molecular data. And what we found is it gave us really a whole new picture of species diversity of Caribbean fishes. We sunk a few species along the way, but mainly what it did by integrating these two data sets was show that there was a lot more cryptic species diversity than we had previously recognized. And then in 2010, I was invited to go down to Curacao in the Southern Caribbean to make some test dives in a brand new state-of-the-art submersible. And I had just spent the past 10 years, studying Caribbean reef fishes, so I thought I had a pretty good handle on species diversity. But here at 160 feet, and also at 300, 400, down to close to 1,000 feet, I was seeing a lot of fish species that I didn't recognize. And these fishes are inhabiting what are known as tropical deep reefs. They extend from about 120 to at least 1,000 feet, and globally these ecosystems are poorly explored. That's because they are too deep to access using traditional scuba gear. The limit's about 120 feet. And they're really too shallow to be of much interest to really deep diving subs. So if, like James Cameron, you have a submersible that goes to 36,000 feet, you're not stopping at 300 feet, 600 feet, et cetera. So this zone between about 120 and 1,000 feet has largely been missed by science. So the Curacao submersible, which is the one that I went down to test dive in Curacao, has a maximum depth of 1,000 feet, or roughly 300 meters. So I knew that if I could launch an effort to use this sub, that we could pay some attention to this poorly explored deep reef zone. And one of those funny life coincidences, I had been at the Smithsonian for many years, and there had never been any startup funds available for new research projects. But at the time I get back from Curacao, having made some dives in this sub and getting excited about it, the Bill and Melinda Gates Foundation had just given the Smithsonian $10 million for research, and it was specifically for startup projects or also for mature projects. So I was able to get a grant and take a team of scientists down to Curacao and launch what we ended up calling the Deep Reef Observation Project, or DROP for short. And that's that was in 2011. We've been going for about 8 years, and the project now includes at least 40 Smithsonian scientists, staff, students, educators, fundraisers, lots of people involved. But the aims of DROP are to explore biodiversity, ecology, evolution, and long-term changes on Caribbean deep reefs. The Curasub submersible is based in Curacao, which is way down near Venezuela. You may be more familiar with Curacao as the home of blue Curacao liqueur. This is the sub. The owner is a private citizen in Curacao. He owns the Curacao Sea Aquarium. He's just a treasure hunter, explorer at heart, and he used to do deep reef diving with mixed gas and rebreathers down to 100 meters, 300 feet or so. And then when he turned 60, either he or his wife decided he was too old for that, and he got a couple of his business partners together, and they had a $2 million submersible built for his private use. So that's Dutch. He's the owner. And the Curasub holds 5 people, so 2 people lie on their stomachs on these benches in the front and look out of this front bubble. The pilot sits on a bench between their legs, and then behind the pilot is another bench where 2 people can sit, and there's a porthole on either side. And then the pilot's, navigating using these screens, and the depth gauge there is in feet. So I mentioned that Dutch really bought this sub for his own private use, but to pay for it, he he made it available for tourists. So actually, it's still being used that way today. Any of you can go down to Curacao and make a dive in this submersible, and the cost varies by how long you want the dive to be and how deep you want to go. But when Dutch started, when he got this sub and he started making these dives and he got below the depths he'd been scuba diving to, he started seeing all this marine life that he didn't know what it was. And so he contacted one of my colleagues, Jim Van Tassel, who had visited Curacao many years ago. And he told Jim, he said, "I've got this sub, I'm seeing all these sponges and corals and fishes and crabs, I don't know what they are. I want to work with one institution that has scientists that know all of this stuff." And so Jim said, "Well, call the Smithsonian." And Jim gave him my name, but didn't tell me he gave him my name. So I'm sitting in my office in the Smithsonian in November of 2010, and the phone rings, and I pick it up, and this voice on the other end says, "Carol, this is Dutch in Curacao." Like, I'm thinking, who the heck are you? But he proceeds to tell me about this brand new state-of-the-art sub that, that he's built, and all these, fish he's seeing. And 45 minutes later, we end the call, and he said, "Get yourself down here. I'll put you up. I'll put you in the sub, and I'll feed you a bad hamburger." So that's basically what I went down to do, to make those test dives with him. And I couldn't have been more impressed with this sub is just a gem. And also I was very impressed with his whole staff. They run this thing like a military operation, so it's very safe. But because Dutch has this aquarium, he had equipped his sub with these hydraulic collecting arms, a high-definition underwater camera, and as I mentioned, it goes down to 1,000 feet. So it turned out to be a fabulous vehicle for research, in addition to just tourism. And finally, this sub offers very easy access to deep reefs, and that's because off of Curacao, the deep reefs occur very close to shore. So this is an aerial view of the sea aquarium. The light-colored water you see is shallow. The dark is deep. The sub is based here. This is called Substation Curacao. They put it in the boat slip here. It comes out of the boat slip, and immediately you can get down to these deep reefs. You can literally, I can get on a plane in D.C. in the morning, fly down to Curacao, be making sub dives on deep reefs by 3 o'clock in the afternoon. It couldn't be any easier in terms of submersible operations. So they keep the sub here. This is the substation. They keep it in this garage, and when it's time to dive, they wheel it out. They lift it with a crane. They place it in this boat slip that was designed especially for it. Open the hatch door, passengers climb in, close the hatch door, out you go, down you go, and again, within minutes you can be at deep reef depths. So in terms of collecting capabilities, the sub has, first of all, it has a basket in the front, and for sessile things like sponges or sea urchins, we can simply scoop them up. This is a suction hose. Diameter is about like that. So things that we suck up go up and around, and they empty into this canister on the outside. This arm also has pinchers we can use to pick up things. For fishes that I work on, they need a little incentive before they'll go into that suction hose. So this hydraulic arm has what's called a quinaldine ejection system. Quinaldine is a fish anesthetic. It's what all the commercial aquarium guys use to collect fishes. It doesn't kill them. It just makes them sleepy, and they start kind of going sideways, and then we can suck them up. If we get fish that are too big for the suction tube, which we often do, we can anesthetize them with the quinaldine and then get them in the front basket, and then we can actually open this hatch door in the back, drive the sub forward, and get the fish back that way. So a lot of different ways that we can collect using the sub. So when we get back to the surface, we take everything that we've collected to a makeshift lab in the Curacao aquarium. And of course, we do the standard, we photograph, measure, take tissue samples. Some of them are for immediate barcoding, others go into our biorepository. And then we ship all the specimens back to the Smithsonian for permanent archival in our natural history collections. By the way, I saw yours this morning. It was just really fun. Had a great time out there. All right, so what are we finding? So we, as I mentioned, have scientists as part of DROP who not just studying fishes like I am, but corals, sponges, crustaceans, mollusks, echinoderms, hydroids, lots of different groups. And so what I thought I'd do is just, first of all, just show you a couple fabulous pictures. There's a professional photographer that was based at the substation named Barry Brown, who is not there anymore, but he just took some amazing images for us of things that we brought up from the deep reefs. And this is a Gorgonian coral with a brittle star intertwined. This is a little Acanthodromia crab. It's about the size of my thumbnail. These are slit shells. This is a living fossil. These slit shells are well known from the fossil record hundreds of millions of years ago. Only a few species exist today, and these are highly prized shells by shell collectors. And these are called tusk shell hermit crabs. So they live in a tusk shell, and they have one of the claws greatly modified so that when the crab retreats into the shell, this modified claw forms a perfect covering to this opening. In fact, when we first collected these, we thought they were the actual tusk shell mollusks because they were closed up. Brought them up, put them in an aquarium, and out crawled these guys. It was incredible because we have specimens in the Smithsonian collection from 100 years ago, but they hadn't been collected any time recently. Nobody ever knew they had any color pattern. So not a new species, but a pretty amazing find. And this picture, which was called The Three Amigos, went global on social media. So what I'm going to do is just tell you about some of what I think are the major discoveries so far in this DROP research program. And the first I just like to call a biology bonanza. And by that, I basically mean a lot of new species. So far, we've discovered over 30 new species of small cryptobenthic fishes. These are just some that we've already named and described. We named one after the Curacao. We named one after the DROP program. So it's been kind of fun. We've had some fun with the naming. We've also discovered a lot of invertebrates. And a lot of the new species of invertebrates have come from bottles that we have found discarded on the ocean bottom. And what's going on here is that octopus, you can see one in this bottle here, are apparently using these discarded bottles as dens. So they go out, they find these micro mollusks, and they bring them into their bottle, their den, and they eat them. And so often we'll, pick up a bottle and it's just chock full of these little shells, but they're all empty. A lot of these are new species, but the scientists who work on them can't describe them because there's no animal in there. The octopus has already eaten them. But just because the mollusk isn't in there doesn't always mean that they're empty. When the octopus gets out of there, apparently the tiny hermit crabs crawl in. And we thought at first that these little hermit crabs that were occupying a lot of these shells in the bottles were just the young stages of big hermit crabs. But our scientist who studies hermit crabs discovered that that's as big as they get. And earlier this year, he described 6 new genera and 10 new species of micro hermit crabs, mostly from bottles. That's the tip of the iceberg, he said. It's an entirely previously unknown fauna of micro hermit crabs. So you hate to encourage littering, but we actually take bottles down now, and we strategically place them, but trying to get octopus to go in there and bring stuff in. But it's also not just things that are inside. One of our scientists at the Smithsonian, Dave Pawson, described a new species of brittle star of the genus Astrophiura. And it occurs on the outside of bottles, down at about 300 meters, roughly 1,000 feet. That's the dorsal view, the ventral view. Interestingly, we've only ever found this new species on bottles or car tires. There it is on a car tire. We've looked everywhere for the natural habitat of it. It's got to be somewhere, but we haven't found it. But this turns out to be a Heineken bottle, and so I thought, maybe Heineken would help fund some of this research. We could name this, Astrophiura Heinekeni. And so I approached them. They sent a team from Heineken USA to the Smithsonian. We did the whole dog and pony show, told them about it. And they do fund research, but it's almost all water quality research, which anybody who drinks Heineken, I think, can appreciate that. So they loved this project, but it was just a little bit outside of their reach, so we didn't get money for them. They ended up naming it Carolee after me, so that was pretty fun, too. And of course, we have new species of octopus. We've collected 7 octopus so far. I had an undergraduate student as an intern last summer, and she determined that 3 of the species, including one from the bottles, represent new species. So we're working out a description of those right now. Okay, so altogether, we have at least 7 new genera and over 60 new species. I think with all the small invertebrates, including the hermit crabs that are still to come, pretty soon we're going to be well over 100 new species. And this is remarkable, but what makes it really amazing is that almost all of this new biodiversity has come from a tiny plot of water. Our study site's only about 1/10 of a square mile, about 3/10 of a square kilometer. And that's because, as I mentioned, the 1,000-foot depth limit of the sub is very close to shore. So the sub comes out here, it turns right, it turns left, and very close to shore it reaches its depth limit. So I think you can see if we're finding this much new biodiversity in this tiny plot of water, and these deep reefs occur in tropical regions around the globe, we've only scratched the surface in terms of understanding what inhabits them. So you're probably familiar with these figures, but it's estimated that 1.3 million species are known. It's estimated that 3 to 10 million species are yet to be described. And the point that I want to make is that in the ocean, a lot of this new biodiversity is going to come from these tropical deep reefs. So these ecosystems definitely warrant a lot more study. And fortunately for us, the owner of the sub, Dutch, bought several years ago a former NOAA research vessel. This thing is called the RV Chapman. It was rusting away in Puerto Rico. Somehow he managed to sail this rust bucket down to Curacao, put about $1 million into renovating it. It is beautiful. He put all these staterooms that have bathrooms and added a crane. So now we can put the sub on the ship and carry it around to other places. So we can really expand our exploration of Caribbean deep reefs. So far, in addition to Curacao, we've been to the nearby island of Bonaire and to the eastern Caribbean islands of Dominica and St. Eustatius. And the dark arrows show some of the places that I'd really like to get to. In fact, I combined this trip here with some meetings tomorrow and Saturday morning with one donor and another potential donor who want to get the ship and the sub to the Bahamas. So that's going to be a pretty expensive expedition. That's a long way to go from Curacao up there, but I'm really excited about it. There was some sub-diving in the Bahamas in the 1970s and the early 80s by the Johnson Sea Link subs that were based at Harbor Branch in Florida. But that was before we were collecting tissues for genetic analyses, so it's like we need to start all over with anything they collected. So anyway, hopefully that's going to go off in 2019. We really want to get to Cuba, somewhere in the Western Caribbean here, and Trinidad, Tobago would be great. But anyway, we'll see. The subdiving, when we're going right off of the Curacao Sea Aquarium, is $5,000 a day. That's for 2 3-hour subdives. That's as cheap as subdiving gets. And that's because it doesn't require a ship fuel, ship crew. When you add the Curasub to its mothership and move it around, it's $18,000 a day. So I was telling the students at lunch that I tell people I used to do science, now all I do is try to find money to get this sub moving around the Caribbean. So if you happen to know anybody, let me know. All right, discovery number 2, a new ocean faunal zone. So prior to our work on the deep reefs in the Caribbean, this is pretty much what was known about reef faunal zones that occur above the deep sea. You have the shallow coral reefs, highlight, all the common things you're used to seeing. And then below that, a zone called the mesophotic. Meso is mid, so mid-light. And the mesophotic is defined as still having enough sunlight that photosynthesis can occur and you can have reef-building corals. Maximally, that's 150 meters, roughly 500 feet. Usually when it's actually been measured, it's a lot less than that. So very little attention had been given to tropical depths below the mesophotic, and the few papers that actually mentioned those depths in the scientific literature suggested that the mesophotic transitions directly into the deep sea at about 150 meters or 500 feet. So we set out to answer that question, and of course we're using the Curasub to dive to 1,000 feet, roughly 300 meters. And we found that no, in fact, the mesophotic does not transition directly into the deep sea, that there's a previously unrecognized zone, which we named the rarophotic for scarce light, that is home to a unique reef fish fauna that are very much like reef fishes and not deep sea fishes. And these are the data that we used to recognize this zone. We had about 4,500 depth observations of about 71 deep reef species. This is a cluster analysis. The mesophotic had already been divided into an upper and lower mesophotic zone. That was based on both corals and fishes. Our data from Curacao completely support that. And then the rarophotic could have been divided a number of ways, but there were 2 zones that fell out, and we named those the upper and lower rarophotic. So the fishes inhabiting the rarophotic, this is just a sample of them, and again, they not only look similar to shallow coral reef fishes rather than deep sea fishes, but it turns out that they belong to families, all these here in yellow, that are predominantly shallow reef families. Only 3 species that we found in the rarophotic actually belong to families that are primarily deep sea inhabitants. So we went ahead and named the shallow reef zone the altiphotic highlight, just to complete the naming classification. And as we start moving the sub around the Caribbean to other places, we're really going to be looking at defining these zones in different places. The absolute upper and lower boundaries may change based on temperature, light, and other things. Okay, discovery number 3. Deep reefs could play a role in the survival of shallow reef life. So you're all familiar with the fact that reefs are in trouble globally. This is particularly the case in the Caribbean. A lot of reasons for this, but the one I'll talk about is temperature. Rising sea surface temperatures, of course, stress the algae that live in corals, and they, cause them the algae to die, the corals bleach, and so that's one of the problems facing shallow coral reefs. Although there are a lot of long-term data sets on sea surface temperatures and temperatures on shallow coral reefs and estuaries, there are no long-term data sets on what's happening on a shallow to deep reef profile. So we are doing this off of Curacao. We have 11 of these oceanographic temperature loggers. They're only about this big, about that big around. We would set 11 of them out between 50 and 800 feet off of the sea aquarium. That's one in situ at 400 feet. They take temperature once every minute. After a year, we bring them up, download the data to a computer, check the battery, replace it if necessary, and put them back out. So as I speak, they're out there recording year 6 data, and we'll pick them up in November. So this is the beginning of a globally unique long-term data set. So this is what all of the temperature data look like in year 1. All of the 11 depths plotted together, shallow to deep. And you can see a lot of mixing at the surface. And then, not surprisingly, it gets colder as you go down. A lot of the same up and downs, you know, whether you're on shallow reefs or deep reefs. One of the things we're doing with this is that we are trying to determine the upper and lower depth ranges of as many fish species as possible, and then we're overlaying this temperature data on those depth ranges to try to come up with each species' preferred temperature regime. One of the things we want to know is, are species moving deeper in response to warming surface waters? So we know that at higher latitudes in both the northern and southern hemisphere, fishes and invertebrates have been documented moving further north and further south, presumably in response to warming surface waters. But if you're a fish living in Curacao, you might have to go pretty far north or south to get to cooler waters, but you only have to go a short distance deeper. So unfortunately, we don't have the historical temperature data and depth data on these fish species to know whether this is already happening or has already happened. But there's another way to look at it. So if you look at this graph, this is depth on the bottom, surface to deep, to 1,000 feet. And this is temperature, warm to colder here. And the black line shows the thermal profile in the southern Caribbean off of Curacao. And the red line shows the temperature profile in the eastern Caribbean from when we went to Dominica and St. Eustatius. And you can see that at just about any depth, the water is a lot warmer in the eastern Caribbean. So if we look at how the fish species are distributed in these places, we see that, sure enough, the fish species are living deeper in the eastern Caribbean where the water is warmer. Same for this here. These are very preliminary data. As we move the sub around on its ship, this is really what we're going to be looking for in addition to new biodiversity, and that is just documenting these temperature profiles and the depths of the species. So you may have seen a paper that just came out in Science recently by Luis Rocha and some of his colleagues that we're looking at the deep reef refugia hypothesis. Deep reefs have often been suggested as a place, as ecosystems that might save shallow coral reefs. And they argued that this was not going to work because in order for the deep reefs to serve as refugia for shallow reefs, you have to have the same species that occur from shallow down to deep. So if the shallow ones disappear, the deep reef populations can reseed the shallow reefs. And what they found, and our data support this, is most fish species that live in the shallow reefs are not deeply distributed. In fact, the fish just break out into different zones, the shallow reef, the mesophotic, and the rarophotic. But what I think is that there's another way to look at this deep reef refugia hypothesis, and that is if fish species and invertebrate species are moving deeper, you could have entire shifts from the altiphotic to the mesophotic, mesophotic to the rarophotic. And that's what we're really trying to investigate as we move forward going around the Caribbean. Now, I mentioned that there are no historical temperature and depth data to know if fish species are already moving deeper, but there is historical evolutionary information that tells us that in the past, shallow reef fishes have moved deeper. My former postdoc, Luke Tornabene, who did his PhD at Texas A&M Corpus Christi, a group of us looked at evolutionary relationships of these small fishes called gobies. And you don't need to be able to read what's on the tree, just know that the blue lines represent shallow goby species and the red ones represent deep reef goby species. And as you can see, between about 10 and 14 million years ago, there were 4 separate transitions of shallow reef species onto deep reef species, or onto deep reefs. And in 3 cases, the deep reef species radiated so that we now have entire evolutionary clades of these deep reef gobies. And amazingly, every one of these deep reef species are new species that we discovered with drops. So this was thought to be an entirely shallow water group of fishes, but obviously it's not. So Luke and some other colleagues and I are now doing the same kind of study on other fish groups. And what we're really hoping is that we can get some scientists interested in doing the same kind of thing with invertebrate groups. So if we can see similar patterns across multiple taxa, then we might be able to make some sense out of what was going on in the past in terms of these depth transitions. Okay, discovery number 4, arms and farms can be used to monitor deep reefs as well as shallow. I don't know if any of you are familiar with ARMS. They're autonomous reef monitoring structures. They're basically little reef condominiums made up of PVC plates stacked on top of one another. The idea is you put them out on the reef, you leave them for a couple of years, all kinds of planktonic stages of reef life settle on them, start growing, and you go down with a milk crate lined with fine mesh, place it over the top, wrap it with a bungee cord, bring it to the surface, and you've got a sample of much of the small cryptic stuff living on a reef, and you don't have to damage the reef to get it. Also, very importantly, every one of these is constructed exactly the same way, so it gives you the standardization that you need to be able to detect changes from place to place and over time. So these ARMS units have been set out on shallow reefs around the globe. There's hundreds of them out there. NOAA really started this about a decade ago. But nobody ever put them on deep reefs, and we weren't sure that we could use the sub to do this. But we ended up taking, the first year, just 12 of them to see if we could, deploy them with the sub and retrieve them, and that worked. And we ended up putting 36 of them out at these 4 different depths. To deploy them and retrieve them with the sub, we had to modify the arms just a bit to create a base plate with these U-bolts. And then I think the next slide shows the milk crate. Well, you can see we're just dragging them off of the, we load them up on the basket here, drag them off with the hydraulic arm and put them in place. That's what they look like at 300 feet after a year. And then, so here's the milk crate, and it has these latches. So when we go down to retrieve them, we take the milk crate in the arm, place it over the arms, arm over the arms, and then you have to use the basket of the sub, or the sometimes just the arm, the hydraulic arm will work to press down on that and latch the milk crate onto the arms unit. But it's amazingly successful. We've been coming up with these arms units, and we've left some of them out for 2 years, some 3 years, some 4 years. We're getting ready to go in November and bring up the last 12, and they've been out there 5 years. When we bring them up, first of all, we take the modal stuff, the shrimp and the crabs, or swimming or crawling around. Sometimes some little fish, not very many fish in these. And then we take the plates apart, and we subsample everything that's growing on the top or bottom of each plate for barcoding, and then we scrape everything off of these plates, put it in a blender, and then we use the metabarcoding or next generation sequencing to process these samples, and that gives us a DNA sequence for everything that was living on that arms. Truly extraordinary technology that's expensive and time consuming, but it's pretty amazing. So we haven't published anything on this yet. We're still in what we call the biodiversity discovery phase of this. And as you can see, these are the 4 different depths. We have the arms, and all of our species curves are still going up. We're looking for them to plateau. But even if they don't, after we bring the next group up in November, we're switching from a biodiscovery phase to a monitoring phase. So next year, we will deploy 12 of them. They'll stay out for 5 years. And we'll, bring them up and then put them back out. So it'll be every 5-year kind of monitoring phase. One of the postdocs at the Smithsonian took the arms and he modified it to make it a fish-specific arms, or a FARMS. And basically what he did was got rid of most of the PVC plates and put different sized PVC pipes in there. We tested this with the sub. We put 3 of them down last year, brought them up, and got a goby species that was new. We'd never seen after 8 years of sub diving there, so got kind of excited about these. But we ended up modifying the design a little bit, and when we go in November, we're going to put 4 of the new ones down. And hopefully we will also ultimately use these in addition to arms for long-term monitoring on a shallow to deep reef profile. The Smithsonian has what's called the Global Earth Observatories, or Marine Geo. And this is going to be a network of coastal marine monitoring sites that meant to mirror what we've already been doing for decades in the terrestrial world. We have a program called Smithsonian GEO, Global Earth Observatories. That's been phenomenally successful, plots all over the world that people have been monitoring for decades. But nothing similar had been done in the marine realm. The first, the Smithsonian marine stations on the Chesapeake Bay in Florida, Belize, and Panama are the 4 initial sites in this network. These are 2 of the partner sites in development, and these are some of the possible future sites. And as you can see, Curacao is there. We're fighting hard for it because it's the only one of these sites that allows us to not just monitor the shallow, but the shallow to deep reef profile. It's going to take some long-term funding, and right now we're trying to work with the Curacao government on that because it won't be a lot of money that's needed, but some will be needed. So we'll see how that goes. Okay, last but not least, discovery number 5. Invasive lionfish are bad. Well, you all know that, but we're looking at it in a little bit different way. In Curacao and other places we've been with the submersible around the Caribbean, we have found these lionfish down as deep as 800 feet or more. In their native Indo-Pacific, they tend to inhabit the shallow reefs. In fact, FishBase, I think, says that the maximum depth is 30 meters, roughly 100 feet. So they're definitely living deeper than that in their invaded territory. And also, they can occur in huge numbers. This is just a few of a herd of about 200 of them that we filmed off of Bonaire. In fact, when we took the ship and sub over to Bonaire, we had the head of the Bonaire Park Service in the back of the sub with us, and she was telling us about this lionfish eradication program they have in the shallow reef. So if you go to Bonaire, most people go just for scuba diving and snorkeling. That's pretty much all there is to do in Bonaire. It's a small little island. But they have pretty much wiped out all of the lionfish in the shallow water. As soon as we got down below scuba diving depths, lionfish, that was the most common thing we saw. And I was telling Kirk earlier, typically these lionfish are reef inhabitants like this. They'll, just spread their fins and they'll kind of blend in with the reef. A lot of the 200 or more that we saw in Bonaire were on sand bottom, and that's not even their natural habitat. They were just anywhere. So the poor lady from the Park Service was in the back of the sub in tears, and she thought they had wiped out lionfish, but they'd only wiped out the ones that are living in shallow water. I was working today on a proposal to further investigate the deep reef lionfish because we know that these things are voracious predators. They'll even eat one another, apparently, but mostly what they eat are other small fishes and invertebrates. But of course, that's exactly what all of our new species are on these deep reefs. So our concern is that these, predatory lionfish and all their huge numbers are down there on these barely explored deep reefs, gobbling up biodiversity before we even know it exists. Unfortunately, and the reason I call lionfish bad is that's exactly what Luke and I found already happening. We described a new goby species, Palidogobius incendius, and we have video of lionfish eating these gobies. So this was the first record of invasive lionfish preying on undescribed biodiversity. This is what the fish looks like. It's tiny. The good news is anytime you see it, it occurs in these big schools, and we've not only seen it in Curacao, but in the eastern and video from the Western Caribbean as well. So probably not too much of a threat to its existence because it's very abundant, but it was the first record of undescribed biodiversity being eaten by these lionfish. So we wanted to focus more on what these lionfish are eating on these deep reefs. So we came up with a plan, which was to go down and collect lionfish on the deep reefs the way we collect other fish. Just squirt the anesthetic at them, let them get sleepy, and then most of them are too big for the suction tube, but suck them, get them in the basket and get them back that way. Unlike any other fish that we've targeted on the deep reefs, the lionfish would not succumb to that anesthetic. I mean, if you've ever seen lionfish or speared them, you can get right up to their face. I mean, they just sit there, and we could do that with the anesthetic. Give them a big squirt of this stuff, see them, taking it in, and then they would just start swimming. And it reminded me of the Energizer Bunny. They just keep going and going and going and never stop. And they would never stop long enough that you could either try to, scoop them up or try to hit them again with the quinidine. So plan A did not work. So Bruce, who's one of the sub-pilots and the chief engineer of the Curragh sub, came up with Plan B, which he said, these things are so easy to spear in shallow water. Let's spear lionfish with a submersible. So he rigged up this pole spear on the front of the sub. We had to just have the pointy end covered with styrofoam while we're on land. But the plan was we'd go down on the deep reefs, find a lionfish. This robotic arm has a little hook, and we could bring it over and pull the trigger on the spear and fire it. So we got down to about 400 feet, and that, ladies and gentlemen, is the first lionfish ever speared by a submersible. But these things are amazingly hardy, and even when the spear goes all the way through this fish, they can still wriggle off and swim away. It was unbelievable. So we learned that once we got them on a spear, you can see the spear tip through here. We had to go down, take the sub down to the bottom, pin the fish against the bottom, and then squirt them with quinaldine until they really just knocked out. And then we could pull them off the spear and scoop them up with a basket. And we've gotten 4 lionfish that way. I mean, it's not easy. So the proposal I was working on today was to get funding to do a lot more of this, but they're rigging up a much better spear system that'll hopefully, really kill these things upon impact. But anyway, so stay tuned about that. And the last thing I'll say about lionfish is that if you have not tried eating lionfish, it is delicious, and that's the best thing we can do in terms of controlling their numbers. We eat it as sashimi all the time when we're in the field. It's great as ceviche, but pretty much any way you can think about cooking it. It's just like any white flesh, like black sea bass or grouper almost. It's really, really good. And while I'm on that, in terms of eating sustainable seafood, I just wanted to point out that in 2002, a colleague and I published this book, One Fish, Two Fish, Crawfish, Bluefish, the Smithsonian Sustainable Seafood Cookbook. And this was really an effort to, it was first an educational effort for myself, because I grew up in coastal South Carolina. We catch all our own seafood down there. And then I moved to graduate school at VIMS at Gloucester Point, Virginia. We caught all our own seafood there as well. When I moved to DC, I had to start buying seafood, and I realized I didn't know what I was buying, where it was from, was it a good choice from an environmental perspective. So I just set out to answer those questions for myself, and I couldn't believe what I learned about how poorly managed U.S. seafood species are. So I looked, instead of telling people what not to eat, I researched ones that are fished or farmed in a sustainable or environmentally sound manner, sent that list out to chefs all over the country, and they sent recipes in. So it was 150 recipes from over 100 chefs. And so I just brought a copy to leave here. And Kevin, I'll just leave this in your office. But it came out in 2003, so not all of the fisheries statistics and stuff, of course, not up to date anymore. But the seafood species are largely still the ones, you should look for, and the recipes are, of course, timeless. And the other thing I wanted to leave with you was, I didn't do this specifically for A&M, but I was just in Curacao. We were making some dives. And I did, we take these styrofoam cups and we write on them and put them on the outside of the sub, and then when you go down, they shrink. So the one thing I like about this one is it said rarefotic or bust. So this one went to about 700 feet, didn't go all the way to 1,000, but anyway, little memento. Okay, I'm happy to stop there. I have about a 5-minute video if you'd like to see it. It just takes you through the the different zones. Let me just say while this is loading that I had a student intern, and I just gave him, just do what you want with this. Let me see, how do I make this full screen? Is it here? There we go. So some of the footage from Bonaire, some from Dominica. If it doesn't say anything, it's from Curacao. But that's, a camera over the sub as the sub is descending. And this is in meters. Roughly multiply it by 3 if you want to, I'm sorry, it's the other way around. No, roughly multiply it by 3. But I just want to point out, this is shallow reef. This is alphiphotic. And you can see in Curacao, there's a lionfish, by the way. The coral is still really nice down there. They don't get hit by hurricanes very frequently, so the coral is in pretty good shape. This is a beautiful expanse of reef-building coral in the mesophotic zone off of Curacao. We're in Dominica now, about 70 meters, and this is a very typical mesophotic scene. There are some hard corals here and there, but lots and lots of sponges, lots of fish diversity, a lot of similar fishes, some even same as you see on shallow reefs. Those are common name? Yeah, flying gurnards, yeah. And what's funny about this is normally when they're disturbed, they, really put those pectoral fins out, but they didn't do that. These sea turtles, if you look at the maximum depth that was supposed to occur, it's like 20 meters, and that was down about 80 meters. So there's a herd of lionfish there, and this is a really typical scene on the deep reefs, where you see one lionfish, you're going to usually see a lot of them. There's some more in the back there. 85 meters off Dominica, we're still in the mesophotic, and again, you can see, in this case, the same angelfish as you would see on shallow. I want to stop this just a second if I can. Because I was telling some of the students last night that in Dominica, this is an island that has some 300 rivers on it, and it rains all the time. And so this sediment, washes from the land onto the reefs, and including the deep reefs. And you can just see the sediment, is just covering a lot of this stuff. So what we found in Dominica is, rather than having a lot of organisms attached to the rock substrate, they were attaching to sponges. So here we just went down with the front basket of the sub, shoved it underneath the sponge. We got like meter-long sponges. We'd bring them up to the surface, and everybody had something to, to look for, whether it was crustaceans or mollusks or fishes or other sponges or corals. It was amazing sampling, but that's what's in the front basket there in the sub. I haven't looked at this in a while, so okay. So that's off of Curacao. That's a new species we haven't described yet. It's in the wrasse family. It's called a red hogfish. So we're about 100 meters here at St. Eustatius, still in the mesophotic zone. As you go deeper, you'll see this over and over. Now we're at 116. By the way, there's a lionfish, and those little things swimming around, that's the new goby. And you can see how tiny, yeah, there they are right there. You see them? Yeah, you can see how tiny they are. But so this is a vertical wall of St. Eustatius, and you can see just covered with life. So these reef-like ecosystems definitely do not stop on the shallow reefs. Okay, now we're in the rarephotic. And these are jackknife fishes related to ones that live on shallow reefs, but this is a deep reef species. And then the main thing you notice when you start cruising around the rarephotic is that you still have a lot of corals, but they're primarily Gorgonian corals or solitary hard corals, or these Gorgonian coral whips. You no longer have any reef-building corals at these depths because there's not enough sunlight. But the habitat can vary quite a bit. Here it's sand. This, by the way, is the sub's downline. That's a big coronet fish. That's when we are in the sub and we go left or right, when we hit that line, we know it's time to go up. That's like the breadcrumbs home. This is called a big eye. It's a very common rarephotic fish. That was just a barracuda visiting from above. Very typical rarephotic scene where just a reef-like ecosystem, but these Gorgonian corals and some sponges. That's just a big tube anemone. There's a big conch with a hermit crab in the front basket there. That's another very common rarephotic fish species, a boarfish, B-O-A-R. That was a conger eel that we squirted with quinaldine. We didn't know what it was. We just saw the mouth sticking out, and so we didn't end up collecting it because we didn't need it. But it was a nice sight. That's a big flatfish, big flounder, and that's a bottle there that was empty, so we didn't pick it up. But you can, when you're looking out of the front bubble of the sub, everything looks a lot smaller than it is because of the sphere. This is, by the way, hydrothermal activity in Dominica. No real, vent systems at these areas, but that's a sea robin. We have a new species of sea robin that we haven't described yet. This fish we tried to catch, it's a batfish, big one, like this big, but we had hit it with a quinaldine, but it wasn't knocked out enough. And that's the deep reef relative of the deep sea anglerfishes. This is called a deep sea toad, and unlike the deep, the really deep ones, it has color. It's orange, yellow, and there are moray eels down even as deep as 300 meters, and this is Gymnothorax matarensis. It's we've seen quite a number of these down close to 300 meters. And this eel apparently is using the lights of our sub to forage. That's a new species of black coral that we haven't described yet. There's a scorpionfish. This is a vertical wall off Curacao that goes between about 200 and 300 meters. I don't know if you're familiar with orange roughy. Some of the professors maybe. It used to be popular at seafood markets, but way overfished. And this is one of the orange roughies. And that's a fairly common rareophotic fish too. There comes an octopus crawler at almost 300 meters here, maximum depth of the sub. And you can just see how those things change colors instantly when they hit different backgrounds. And this is a rareophotic, a deep reef snapper. Again, right at the maximum depth of the sub. But again, I mean, that's not a deep sea fish. That's a fish that's related to shallow fishes. And the fish that the sub just went over is just a lizard fish. And that, again, is another species that belongs to a primarily shallow water family that lives on shallow coral reefs. Okay, thank you, and I'll be happy to answer any questions. Who wants to go diving? Okay.
How long can you stay under for?
In the sub?
Yeah.
Yeah, all right. The question is, how long can we stay under? When the batteries are completely fresh and charged, that sub has like over 24 hours of capacity. So if you got stuck down there, you'd be okay, for a while. But they tend to not run the sub any more than 8 hours at a time. It really runs, apparently when you keep it going longer than 4, 5, 6 hours, it really runs the batteries down a lot. But typically we do 3 to 4 hour dives so that we can get 2 in one day. And you really don't want to be, I was in the sub one time for 7.5 hours. It's just too long. There's no bathroom, and you're either lying on your stomach, propped up the whole time, or you can, if you only have three passengers and a pilot, you can switch places pretty easily, so somebody can change from laying on their stomach in the front to sitting in the back. You sit. So the hilarious thing about the 7.5 hour dive, it's not really hilarious. It's sad, but so the owner, Dutch, was just gung-ho to explore somewhere. He didn't have his ship yet, and he was just gung-ho to explore somewhere other than right off of his aquarium. So he got when we're in the sub, there's a surface boat above us, and we're in constant radio contact with them, and every 30 minutes they call down for life support. So the pilot reads all the gauges, and they record all that. So it's a very safe thing. But Dutch said, well, let's get the surface boat's called the Johnny Boy. So let's get the Johnny Boy to tow the sub down the coast a bit so we can dive in someplace new. And so we did that, and we ended up in this fabulous place. I mean, it was just loaded with fish and things we hadn't seen, and we spent hours collecting there. But we filled up the front basket with stuff. When they tow the sub, they tow it backwards so that the front bubble isn't getting beat up. But they couldn't tow us home backwards because all that stuff in the front basket of the sub was going to come out. So we had to motor back, with the sub, and it took us 2.5 hours or whatever to get back to where we were. We get back, and when the sub comes into the little platform there, they lift it with the crane and bring it out, and then is when we take everything out, of the basket, and we take that canister off. Dutch, who loves to drive big machines, but he's not nearly as good at it as some of the younger people, jumps up on the crane to lift the sub out. As he's lifting the sub out, he bangs it against the wall, the canister that we've just collected 7 hours worth of stuff. falls open, everything goes into the ocean. So we jump into the ocean, we get anything we can grab, cups, I mean, and we're just throwing stuff, and we lost half what we let the fish just swim. And it was like, oh my God, I can't believe it. We were dying. I mean, we had to go to the bathroom so bad. It was Friday afternoon. I think it was like 6:30, and the sub crew was, like ticked off. They wanted to go to happy hour. So, so we found that, what we pay for is 6 hours a day, and we try to do 3 hours dive. Now, he doesn't, it's hard to tell exactly how long it's going to take you to get back from a certain point, so he doesn't clock us to the minute. But yeah, so those are completely tolerable. But 7, 8 hours, that's tough. Yeah.
Does making it to 1,000 feet make you want to go deeper, or does the diversity increase?
Well, the question everybody heard it is, does this make me want to go deeper? And I didn't mention, but I had the chance in 1998 to do some submersible dives in the Galapagos Islands in the Johnson Sea Link sub, which goes to 3,000 feet. And I found that for the fishes that I'm interested in, this is about the maximum that I want to go, because even at Curacao, when we hit that 300-meter, 1,000-foot mark, we're starting to turn into deep sea, stuff. So I am excited as heck about staying in this range. Now, that depends on what you're studying, because even in Galapagos, even at 2,000 feet, we found, and even at 3,000 feet, I mean, there's just a ton of the diversity definitely decreases, but there's still a lot of new stuff that we don't know about. And we found new species at 1,000, 2,000, 3,000 in Galapagos, and as you keep going down, it's gonna be the same. But the diversity definitely decreases, and it's very unusual on the deep reef to land on a bottom where you cruise for any length of time without seeing life. I mean, if nothing else, you're going to see hermit crabs walking across the bottom, or sea urchins or something. In Galapagos, at 3,000 feet, we'd often land on a sand bottom, and we could go for, I'd say, half a mile without seeing anything. So that's not that much of a distance, 1,000 to 3,000, and it changes a lot. So the diversity definitely drops off. So Dutch is so eager to go deeper at his own place because he's just curious. He wants to see that. I was telling Will, who was here earlier, that because Will has trained to use the deep workers. The deep workers, you have a trained pilot and then one passenger, and you're running the thing yourself. And one of the deep workers goes to 2,000 feet. So Dutch's birthday is in October, and he's thinking about getting one of the deep workers from Vancouver, Canada, to ship it down to Curacao so he can go to 2,000 feet just to say he's done it. And I said, you do it, give me the video, give me the temperature data, and that's all I really want. Because I really want to, because his sub is limited to 1,000 feet, we couldn't definitely define the lower boundary of the rarefotic as 300 meters. That was the maximum depth of the sub. We can say that at that point we're starting to see species that belong to deep families, but you need to go below to be able to define the lower limits. So that would be really my main reason for wanting to go deeper. Yeah.
I don't know if Kevin mentioned, but we've been teaching a study abroad class at Dominica for about 30 years.
Yeah.
And after Hurricane Maria, there was some initial news that there might have been really significant reef damage due to the volume of fresh water and sediment. Have you heard anything since Maria?
Oh, we tried so hard to get Dutch and, to just jump back on the ship with the sub and go right back to where we were. So, because we have hundreds of hours of video from where we were, and if we could have done a rapid assessment afterwards. So I don't know, but my guess is it's not good. While we were there was so much rain that there were times where the sub was coming back up to the ship, at the surface, and logs were coming out of the rivers, and we're like, this is a plexiglass sphere. If one of these logs slams into the sub, we're done. So it was really frightening. So they really got hit hard, and I guess I can't remember the hurricane that hit them a few years before that, where entire landslides, the houses and everything went into the ocean. Yeah. Oh, it wasn't. Okay, right. Yeah. So. But no, and I think NSF does have one of these, rapid assessment grants you could get. Smithsonian employees, by and large, cannot apply for NSF because we're quasi-federal.